Microorganisms in gene therapy. So what is gene therapy? It's an experimental technique aimed at preventing or resolving genetic disease by directly targeting the defective genes. A few approaches are being tested. The first of these is replacing a mutated gene that causes disease with a healthy copy of that gene. The second is by inactivating or knocking out a mutated gene that's functioning improperly. And the third is introducing a new gene into the body to help fight a disease. Gene therapy has multiple approaches, and different vectors are used to transport corrected versions of genes into cells. Different approaches include nucleic acid approach. This is done a few ways. The first is using antisense DNA and RNA. This approach uses Watson-Crick base pairing to block gene expression in a sequence-specific fashion. The second is ribozymes, which are antisense RNA molecules that have catalytic activity and function by binding to their target through antisense sequence-specific hybridization, and then inactivate it by cleaving the phosphodiester backbone at a specific site. An advantage of using ribozymes are that a single ribozyme can inactivate a large number of target molecules. However, ribozymes are RNA, and therefore susceptible to degradative enzymes like RNase. There's also protein-based approaches. The first of these is transdominant negative proteins, or TNPs. These may inhibit because the mutant competes for an essential substrate or cofactor that's available in limiting concentrations. The second is single-chain antibodies, which work by using a heavy chain leader sequence that targets the intrabody to the endoplasmic reticulum and rearrange chain variable regions that are connected by a flexible interchain linker. There's also immunotherapeutic approaches, like genetic vaccines, which are a type of nucleic acid approach and attempt to trigger an immune response to native proteins of the infectious agent encoded by transfer of plasmid DNA into cells. The logic behind this approach is to produce a specific cytotoxic T cell response and a humoral response. So how do we get these new genes into the cell? This is done by using vectors, which can act as transport vehicles to deliver the new desired gene to the cell. These vectors are capable of transferring desired genes, but they aren't capable of taking over or harming the cells. These are divided into viral vectors and non-viral vectors. Viral vectors include retroviral vectors, which infect the target cell through interaction between the viral envelope and a cell surface receptor on the target cell. When the virus reaches the cytoplasm, it's uncoated and the RNA is reverse transcribed into double-stranded DNA. This new double-stranded DNA is then transported to the nucleus where it's integrated into the genome by integrase. There's also adenoviral vectors, which are used for gene delivery in vivo. The genetic material of the adenoviral vectors is not incorporated into the cell's genome. Instead, it is left free in the nucleus and is transcribed as if it were just another gene. The problem with this is that the loose DNA is not replicated with the rest of the cell's genetic material cell division. So, progeny of the cell won't receive the extra genes unless adenovirus is readministered to these progeny cells. And here is an animation illustration of adenovirus vectors being used. Herpes simplex virus vectors are also viral vectors. These carry plasmid instead of viral genome, so no virus genes are being transported. They have a high capacity and allow for transport of long DNA sequences, and they enter by cell fusion. The problem with these are that expression of the new genes tend to stop after 1 week. And the last of the viral vectors are adeno-associated virus vectors, which are small viruses with single-stranded DNA. They insert the new genetic material at chromosome 19. The disadvantage of these are they are difficult to produce and carry small amounts of DNA. However, they are non-pathogenic, so there's no immune response that will remove the virus or cells that have been treated. There's also non-viral vectors. They are less efficient than viral vectors. The two main types of these are liposomes, which are the most widely used non-viral method. They are cationic liposomes and interact with negatively charged DNA to form vesicles. They also interact with the negative charge of the cell membrane. The cationic vesicle fuse with the cell and surface and deliver the DNA directly across the plasma membrane. The second are gene guns, which DNA is loaded onto gold beads and injected into the cell with a helium gun. This can transfer DNA into non-dividing cells. So how do you know if gene therapy is the good solution? It may seem like the ultimate solution to many diseases. However, it's not a molecular band-aid that will automatically fix any genetic problem. There are many disorders and medical conditions that can be potentially treated with gene therapy, while others aren't suitable for this approach. So what are some good candidates for gene therapy? There's germ cell or somatic cell, or the ease of isolation of cell due to accessibility of the tissue organ targeted. So, treating acquired and inherited diseases like cancer, cystic fibrosis, muscular dystrophy, and rheumatoid arthritis are good candidates. There are possible issues with this that include the immune response to the vectors, both bacterial and viral. As we know, fighting infection by bacteria and viruses is among the key functions of our immune system. So to prevent this from happening to the viral vectors, we need to begin to merge some of the qualities of the viral vectors with non-viral vectors to generate a safe and efficient gene delivery system. With respect to immunology, viruses still have many secrets to be unraveled. Viral systems that have evolved to escape immune surveillance can be incorporated into viral vectors. So what are some ways this is actually being used? The first is in cancer immunotherapy. A study done in mice was used Salmonella strains to help target specific tumors. So, model tumor antigens like beta-galactosidase or human GP100 were encoded in a eukaryotic expression vector and they were carried by the strain of Salmonella. They were administered orally and then helped protect the mice against challenges with fibrosarcoma, renal carcinoma, and melanoma cells. The second is the inflammatory bowel disease. This was also done in mouse models. An administration of a recombinant Lactococcus lactis strain, which was made to produce anti-inflammatory cytokine IL-10, led to local production of the cytokine and prevented development of colitis. So what is the takeaway from this? Gene therapy is obviously unique because unlike traditional drug-based techniques, which may treat the symptoms, it actually treats underlying genetic problems. In many cases, the goal of gene therapy is to correct the defective genes in the cells, such as epithelial cells in cystic fibrosis and lymphoid cells in ADA deficiency. Gene therapy usually promises to help diseased tissues and organs function properly. However, because these cells are continuously replaced, there has to be either continued therapy or an attempt to target the stem cells. So we first need to develop further the technologies for identifying and isolating these cells to understand their regulation and then to target infection to them in vivo. Here are our sources to help you further understand and relate to gene therapy.